distinguished speaker series. Just before I introduce my friend Wallace, to the people on the phone, if you have any question, since you're on mute, please same time them to me. Oh, I'm Simone Bianco, by the way, and I will convey them to the speaker. Okay, so it's a real pleasure for me to introduce my friend Wallace today. This morning I was thinking about how to introduce him, and I came up with such a lengthy introduction full of spoilers, and so I'm just going to be basic. I'm going to say that he's an electrical engineer by training that is on loan to biology for the time being. Wallace, and in his lab, he studies problems that are fundamental in biology. Why cells look like they do, how do they came about looking the way they do, and how they solve basic problems like measure and count when they live. And so he does this in his lab using a highly interdisciplinary approach, mixing cell biology, molecular biology, engineering, mathematics, physics, and computer science. And so he's also the main force behind a large collaboration in the Bay Area that involves UCSF, Stanford, Berkeley, the San Francisco Exploratorium, San Francisco State. Am I forgetting anybody?
You guys.
And the Accelerated Discovery Lab here at IBM. And on a side note, Wallace is also one of the reasons why I'm here today. There's a long story behind it that dates back to 2012. So if you like me a little bit, buy him a cup of coffee. If you don't like him, he's to blame. Thank you for being here.
All right, thanks, Simone. It's a great pleasure to be here. So I don't come from biology originally, but I've been immersed in biology for quite a while now. And so if I start doing the usual biologist thing and use a lot of jargon and terminology that people don't know what it is, please just stop me and make me explain it, right? Because nothing that I'm going to talk about should be hard to follow unless I accidentally don't explain it. So don't let me do that. And Simone, you're in charge of stopping me, okay?
Right. So
I want to talk about this question of how cells generate different kinds of geometry.
Except Simone already knows these things, so he's not
some,
maybe, yeah.
Some.
So I think a lot of us, including me, when we're growing up, we're taught this is what a cell looks like. More or less this big bag of water full of chemicals. And people will begrudgingly admit that there's various structures inside the cell at the nucleus. But the tendency is to want to grant the cell the minimum possible amount of structure because because we know how to think about biochemistry and molecules, and we don't know how to think about these kinds of structures. So this is what we sort of trained cells look like, but actually most cells don't look like that, and most cells look much more like this. So cells can make, these are all individual cells, they can have incredibly complex forms, and this is true not just for free living cells you might find in a pond or in the ocean, but also for various cells in the body. And you take something like this hair cell in your ear, these structures are precisely tuned in their size and number and orientation to allow you to perceive vibrations. And you ask, well, how can what is essentially a bag of proteins make something this complex? What specifies this structure? And you can see, if you think about it enough, that to make these structures, you've got to solve a number of basic problems. How do you decide how many of an individual substructure to make? Remember, this is all one cell. And within one cell, do you decide to make exactly this many of these structures? How do you decide how long to make them and where to put them? So, and I would argue that although people have been looking at these kinds of cells for a long time, we know essentially nothing about where any of these structures come from. So one reason to study cell geometry is simply because it's a mystery that we have no idea how biology does it, and the idea is if we understood how biology did that, we would actually learn a lot, and we'd be very excited just with basic curiosity to know how that works. There's also potential applications, one of which Simone and I have been talking about a lot, is that at some level a cell is a lot like a chemical factory, where you have all these biochemical reactions taking place to produce various molecules, many of which are useful. But the study of cell biology is really focused entirely on the chemistry of the cell, whereas chemical engineering involves both the chemistry that's going on, but also the physical structure of the chemical plant. And a good chemical engineer will actually design the various reaction vessels and reflux columns in a very precisely rational way to achieve the desired output and minimize off-target products and so on. And the idea is that if we could learn how to re-engineer cells, not only at the chemical level, but also at the structural level, maybe we could do a new kind of biochemical engineering in cells.
I would think there'd be huge healthcare implications like cancer cells, healing cells.
Absolutely. So, I mean, one of the hallmarks of cancer, actually the way cancer still is largely diagnosed is by cutting samples from biopsy and looking at them in the microscope. And the cytologist looks at this cell doesn't look right. And if you look at a cytopathology textbook, it's full of examples of cells that look weird. The nucleus looks like a coffee bean, or the cytoplasm has all these little granules in it. And I've been pursuing this, and I figured, okay, this field would be a great way to learn about these pathways, because they must know a lot about why these things happen, because it's so critical for diagnosis. There's essentially nothing known, and nobody cares. So every pathologist I ask couldn't care less why the cells look the way they look. As long as it's predictive of how to treat it and what the prognosis is, they don't care why it looks that way. But I think your point is that, well, if we understood why they look the way they look, presumably the cells are doing that for some reason, maybe to compensate for chemical changes that happen when you become a cancer cell. So what would happen if you could then tune that cell back to a more normal structure? Would you kill the cell or make it less malignant? And actually, this is how Simone and I first got to know each other, was we were collaborating on a project to ask exactly that kind of question. And I think that's still ongoing, right?
Yes.
But yeah, I think that's a promising avenue.
And you would learn more about the sensitive points on the surface that can be treated or used
Yes.
as a diagnostic.
Yeah, exactly. So, right, so you would learn new ways to treat it potentially that aren't currently being pursued, because they might be different than, you know, everyone's focusing on the mutations that they see in the cancer cell and trying to fix those molecules. But maybe if you retune the container that the molecules are in, you could fix it without having to fix the actual mutation. And maybe you could fix a whole lot of different kinds of mutations by dialing one knob. And then in terms of diagnosis, again, you know, people are mostly focused on the things that they could see using chemical stains that the Germans developed in the 1800s, basically. And we haven't really progressed beyond that. If we knew a lot more about why these structures were the way they are, we might be able to find different set of probes that would tell us more. Question in the back.
I have a basic question. How do you know what cell is? Like when you look at like a hair, for example, how do you know at which point you reach like the minimal cell? Like, how do you know that there is no like smaller cells in the cell, or that you went far enough to determine this is one cell?
Yeah. So the sort of prototypical view is that you have a cell that has an outer membrane around it with one nucleus in it that has the whole genome. And so you would sort of try to define, you would look, basically what you do is you would look at where the nucleus is and then go outward until you find a membrane that doesn't contain any other nuclei. That will mostly work. However, there are examples where, for example, in the human placenta, many different cells will fuse together. And so now you have a cell with lots of nuclei in it. And it's not at all clear whether that should be viewed as one gigantic cell with many nuclei or many individual cells that just happen to be fused together. And plants represent an intermediate case where most plants, the cells are almost separate from each other, but still connected by little tunnels of aqueous cytoplasm. So are they really all one cell? So I think these are I think the basic answer is you just count where the nuclei are, and that tells you where the cells are, because nothing can survive without a nucleus. But there's all kinds of exceptions that are interesting. Question and then question.
This is a comment. It's plant cell, right?
This would be a plant cell, yeah.
So another reason to understand the why on cancer cells is if this is caused by X, maybe you can stop X from ever happening.
Absolutely, right. And so if you knew what pathways were upstream of these, and this is again part of this proposal we're constantly working on, could you actually infer what molecular changes had happened in a cell just by looking at the structure of its organelles without having to actually sequence its genome and so on? And that would be nice because you could use it on biopsy samples using current pathways, current pipelines for getting the material out of patients. Yep. Okay. So I hope I've convinced you that there's a need to understand where the structure of cells come from. And now the question is, well, why don't we already know the answer to this? Because people have been studying cells for a really long time. Lots and lots of people, lots and lots of money. Why don't we know? I think part of the issue is that right now, molecular biology is our main tool, manipulating DNA, watching what happens. And the tools of molecular biology are excellent for determining what the parts of a cell are and how the parts are made. So how are different genes turned on and off? How are the proteins made? What do the proteins look like at a structural level? So we're really good at knowing how to get all the parts. Molecular biology doesn't really have all the tools in place yet for understanding how those parts are put together, because this is now a super molecular question. How do you combine many of these structures onto structures of length scales that are much larger than the length scale of an individual molecule? What controls structure at that level?
Question.
Yeah.
You talked about cells and molecules. Cells make up molecules or atoms make up molecules?
So atoms make up molecules,
And cells make up atoms?
and then the molecules make up the cell.
Oh, yeah.
Right. So basically, a cell is essentially the outer membrane is basically a multimolecular aggregate of fat molecules that make this this nice sheet, because that's their most stable configuration. And then inside that, it's mostly watery cytoplasm full of proteins and RNA and other molecules like that. Yeah. All right. So the question that I really want to try to learn how to address is what determines how these parts are put together. And the issue is that cells are able to build very complicated structures without having a blueprint. Now, you may hear people tell you that the genome is the blueprint, DNA is the blueprint for the cell. And I'm going to try to argue that that's not the case. Because if you think about what a blueprint is, the whole point of a blueprint is that spatial information is explicitly encoded. Right? So the blueprint tells you exactly where to put each piece, how big to make it, what its shape is, and so on. Whereas the genome doesn't tell you any of that. There's nothing in DNA that says put this piece there, or put it there, and make it so big. All the genome tells you is what to make and when to make it and how much to make. So I think a much better analogy for the genome, instead of a blueprint, it's a recipe book that has no pictures. And if you ever cook, a recipe book with no pictures is great if you just want to make soup. But if you want to make something complicated like beef Wellington or some kind of fancy cake, it's very hard to do that without having some explicit information about spatial structure. So I think there's a really fundamental question, which is, if there is no blueprint, what specifies structure? Where does it come from in the cell? And you can imagine various ways to get at that. The approach that we've been taking is to look at an intermediate level. So we talked about the molecular level and the level of the whole cell. In between that, there's a level of these structures called organelles, which are again multimolecular aggregates, which often are bounded by a membrane and perform discrete biochemical functions. For example, the nucleus contains your genome. There are mitochondria, which basically produce the energy for the cell. And all these different organelle structures have their own unique shapes that can vary from cell to cell and that are altered in cancer. And so our approach is to look at this level and say, let's look at each organelle and try to find out, how does a cell control how big it is, the size, how many of them to make, the number, what they look like, so the shape, and also where they are in the cell, the position. And the idea is that if we dissect the cell one organelle at a time and learn what controls these basic geometrical features, can we put that back together and understand how the entire cell gets its structure? This is the idealized view. So, and ultimately, we like to do this for every organelle. For the time being, we're focusing on ones that are easiest to study. And so for the question of size, which is what I'm going to talk about today, the organelle that we decided to focus on is the flagellum or the cilium. And these two words mean the exact same thing. It's like morning star and evening star. It's just two terms people have for the same thing in different contexts. So flagella are linear structures that protrude from the cell, and typically they move to drive fluid flow. So right now, all of you in your airway, there's a constant motion of cilia that are driving mucus up out of your lungs. Otherwise, we would all be drowning in our own mucus. And so whenever you blow your nose, the mucus got there from your lungs because cilia pushed it out. But cilia also perform many other interesting functions, but essentially they're motile organelles. Now they're also known to act as antennas that can sense molecular signals outside the cell. So the reason we study this particular organelle is because it sticks out from the surface of the cell. It's not buried inside the cell, so it's very easy to see. And it's long. It's on the order of 10 microns, so it's a size scale that's very easy to image with conventional microscopy. So it's easy to see, and it's linear. So it's just a movable rod. And the beauty of that is the only geometrical feature that can change is how long it is. So unlike many other organelles that have very complex, intricate shapes, where you might want to know things like the total surface area and the Gaussian curvature and all kinds of other shape parameters, there's really only one thing to worry about with flagella, which is the length. So it's a one-dimensional size control problem.
That implies the cross-section is always the same.
Yes, and that is the case. Exactly. That's exactly what it implies, right? Yeah, so the cross-section is actually set by an organelle inside the cell body that's a template for the structure, and those things are highly invariant, and they're the same for all species and extremely conserved. Yes?
Yeah, excuse me.
Yeah.
Do they lash back and forth, or do they rotate?
It depends entirely on the cell type. So there's situations, for example, the reason why your heart is on one side and your spleen is on the other is because when you were an embryo that was undergoing a very early stage of development, there's a patch of cilia on the surface of the mammalian embryo which rotate around like this. I mean, they're not really rotating the basis fixed, but they're sort of doing this circular whipping type motion. And they always rotate counterclockwise, and that directed rotation, and they're tilted, and ends up generating a leftward flow of fluid over the surface of the embryo, and the direction of that flow determines which side of your body is going to be your left and your right. And if you actually take, so in humans in which those cilia are no longer able to move, half of them have all their organs on the wrong side of the body. And if you take mouse mutants, so I'm getting really off topic, is that okay? I mean, if you take mouse, so I love this question because when we first started my lab, that's what we were working on. And so a guy in my lab, before he joined my lab, found that if you take mouse mutants where the cilia are paralyzed, so they can't do this rotational, they're just sort of sitting there, now half of them will have their organs on the wrong side of the body. He then built a little pump, and if you put the embryo in this little pump and drive the fluid the right way, you can fix the problem. And if you drive it the other way, you can make all the mice have their organs on the wrong side of their body. It's pretty amazing. Now the good thing is, it doesn't actually matter which side of your body your organs are on, as long as they're all inverted. But some people have sort of a mix and match of some are inverted, some are not inverted, and they have all kinds of problems. Yeah. And then there are patients, and part of the way that was first discovered is there are patients who have what's called Cardiganer syndrome, where they have their organs on the wrong side of the body, and they have trouble clearing mucus out of their airways. And so that was the first hint that these cilia were involved in left-right symmetry breaking. Yeah, so anyway, so there are some that rotate like this. The ones in your airway typically just go back and forth on a plane, but they've got to do an asymmetric motion. So they do this power stroke like that. Then they have a recovery stroke where they move sort of asymmetrically, because this is all happening at low Reynolds number. So if you just did a symmetrical forward and backward stroke, you would just move the fluid back and forth. Right.
So these movements are in the moving stream of fluid that they are embedded in?
Yes.
And so this is something of a hydrodynamic issue, is which way they rotate.
Absolutely, and so there's a beautiful set of results that although there are signals, there are positional signals in the embryo that tell the cilia roughly which way to orient. If you don't let them beat properly, they will lose that information. They'll become randomly oriented. If you then apply an external flow, you can make them all line up to the flow. So they're not only generating flow, they're also sensing the flow in some way and then aligning themselves. So the whole thing is a self-organizing system. People try to treat it like an icing model for spin glass, because they can communicate. They've actually measured how far away you can move the cilia, and you can move them many cell diameters away from each other, and they'll still feel each other's effect. So it's a very interesting problem that I think deserves to be actually explored more.
Is there a moral for swimmers about a better swimming stroke they could do, maybe?
I don't know. I mean, it's such a different, kind of, because swimmers have inertia. So, right, so I think, what you want to do when you're swimming is you take the stroke and then you just coast. Right. Whereas at this size scale, you can't coast. Yeah.
That's all.
Because your mass is so small that basically the viscous forces will bring you to a halt essentially immediately. And we've actually looked at this. So in the same organism, we can change the length of the. So this is whole separate. I can show you movies afterwards if you want. So we've been actually watching individual cells as they swim and looking at their two flagella. So you need a high-speed camera because the flagella beat at about 100 hertz. So you need a pretty fast camera. But then we use mutants where we can change the length of the flagella. So normally. Is it okay if I just talk about this? So normally the flagella, they're 10 microns long and they're just the right length. There's a certain curve, so there are motor proteins inside the flagellum that produce a bend. And they're somehow synchronized by unknown means so that they all bend at the same time, so the entire thing does this really nice curvy bend. So the cell starts with this flagella pointing forward and then it bends them out like that. And so that's basically a breaststroke motion. And then it does this recovery stroke where they move very close to the surface. And basically, they're in this immobilized fluid layer because you have the stick boundary condition. And so they come back in this immobilized fluid, so while they're recovering, they don't move the cell backwards again. This is how all small things swim, actually, like little brine shrimp and copepods. But in order to do this, they've got to be the appropriate length. So if we look at cells in which the flagella are too short, for example, half-length, now they aren't long enough to bend all the way around, so they try to bend, but they can't really bend all the way, so they're more like flippers now. And the problem with flippers is you can't swim at low Reynolds number with a flipper, and so if you watch those cells, they just go back and forth like this. It's really remarkable. It's like a perfect example of demonstrating the physics of low Reynolds number motion.
That's great.
Yeah, I'll show you the movies afterwards. I have them on my...
You've shown us just two flagella, but is 2 the normal number, or is
it depends entirely on the cell. Yeah, it depends on the cell type. So
it's a symmetry issue.
Yeah, so this particular organism, Chlamydomonas, it's a free-living green alga, and these cells swim by themselves, and they're trying to swim towards the light. Yes. So they have so they always have exactly 2, and they're oriented something like 170 degrees apart. So it's almost a perfect breaststroke, but not quite. And that's important because they swim on a helical path, so they're swimming like this. And the reason why they need to do that is the way they sense the light is they have a single photodetector, and it's directional. So so over here, you can't see in the picture, but there's basically there's a layer of photodetector molecules, and then behind them there's a layer of opaque molecules that light can't get through. So it only responds to light coming from one direction. And so as it's swimming, it's scanning with its little eye. And then when light impinges on the photoreceptor, it actually changes the motility of one of the two flagella such that it turns directly towards the light source. It's a beautifully crafted machine. I mean, you can see why the intelligent design people like this kind of thing, because it's hard to imagine how this would happen otherwise.
And you can model that perfectly, because the rules are so clear.
Right, exactly.
Yeah.
Yep, you can model, and the beauty is this organism, we have tremendous genetics. So we actually in my lab used that phenomenon as a way to look for mutants that don't put the flagella in the right place, because it only works if your flagella are here and the eye spot is there. And so we look for cells that weren't able to move towards the light, but they could still sense the light and they could still move their flagella. And some of those would have the flagella coming off in the wrong place in the cell. And so they would see the light, but they would swim that way instead of towards the light. Yes, okay, so this brings up the organism. So different cells, like in your airway, typically there's more like 100 cilia per cell. So we use this cell partly because of the fact that it has two essentially identical flagella, so it's very nice for modeling, for imaging, but also these cells, they grow really rapidly. They have certain genetic advantages that make them very easy to work with. Okay, so one seminal result in this organism that was found actually by my postdoc advisor, this paper he published when I was 1 year old, so I feel like I'm following in his great steps. Joel Rosenbaum found that if you chop the flagella off, which you can do by stirring them in a blender or pH shocking them, almost any kind of stress, the flagella will pop off. They will grow back to their normal length, which is about 10 to 12 microns. And they grow back with this sort of, what people said is decelerating kinetics. So this is time and this is the length of the flagella. So they start off growing back very quickly and then they gradually slow down and they sort of asymptotically approach their final length. And if you just look at the slope of this thing and just, if you read out the slope, it turns out that this thing is, that the rate of increase in length is going as 1 over the length, which ends up being important later on. So because of this result, which was, 50 years ago, it was always thought that the cell must somehow know how long its flagellum is. And the question is, well, how would the cell know how long something is that's 10 microns long? So molecules know a lot about the concentration of other molecules, and that's about it. So how would a typical molecular pathway be able to understand or measure length scales of microns and distance?
Is the flagellum moving when it's growing?
Yeah. I mean, initially not. So as it's growing, it's putting in these motors that create the bend. So there's a period where it's not moving, and then it starts moving somewhere around here. Yeah, and we've done a lot of work to try to ask, is the motion involved in any of the control of the growth or length? And the results, I think, are equivocal. So you can make mutants where the flagella are paralyzed, and some of them are normal length, and some of them are too short. And you can also apply artificial fluid flows to the cell, and you can make the length go up or down a little bit. So there's some link between the mechanics of the motion and length, but it's not. We don't really know what that is. We're ignoring it for now. Okay, so the question is, why do you see this decelerating kinetics? How would the cell know when to stop growing its flagellum?
Maybe it runs into barriers, feels itself run into a barrier. No.
Maybe. So we haven't tried that kind of thing. So mostly we grow them in very large volumes where they never see any kind of a barrier. But this fact that cilia can align to an external flow means there's some kind of mechanically sensitive elements in there. And so you can imagine having those things somehow sensing the resistance as it's moving, because they know the elasticity of their own. I mean, the elastic modulus of the cilium is a constant. So if you could read out how much it's bending for a good amount of force you apply, you could back out and get the viscosity of the fluid. And you might say, well, maybe they shorten the lever arm in order to be able to move more effectively. I mean, that's all just completely hand waving. Again, and we're ignoring all that, so forget I said that. But yeah, that's something that we really want to go back and look at.
Just
So,
a question.
yeah.
The frequency of vibration, what is that the function of?
It's not clear. So again, so there's a whole bunch of these motor proteins that produce the bending, and mutations in those can either increase or decrease the frequency. So I'm going to say we don't know. There's not sort of it's hard to put your finger on an obvious pacemaker oscillator. These motors can act like oscillators. So a lot of molecular motors, if you force them to move while pulling on a mechanical load like a spring, they'll move for a while and then let go, and they will actually their chemical cycle will entrain to their mechanical behavior. So at some level, this whole thing is an array of thousands of oscillators that are all mechanically coupled to each other. So it could be something like the famous Strogatz firefly study where all the fireflies blink with their own oscillator, but when they can see each other, then they synchronize. Yeah. So to try to get at what's going on here, I'm just drawing you a simple picture of what a flagellum actually looks like up close. So inside the cell, it constructs these struts called microtubules, which are they're basically protein-based tubes that are very rigid and straight. And so it will make a bundle of these microtubules, which then push out the cell membrane. And presumably, it is the length of these microtubules that determines how long the flagellum is. So this is something that I've been studying for a long time, starting from when I was a postdoc in the Rosenbaum lab, and now working out in my own lab. And what we found was that it was known previously that when the flagellum first assembles, it grows by adding new protein to the end, so to the far end of these microtubules, right? So it's basically building the structure from the end. And what we could show is that even when the flagellum was done growing and it reached its final length, there was still constant incorporation of new protein, this protein called tubulin. New protein was constantly being assembled, and the reason why the flagellum didn't grow any longer is that as the protein was being added on, it was also being taken off. And so there's some machine putting it on and some other machine taking it off, and we don't actually know the identity of these molecular machines that are doing these two things or how they can both be happening at the same time. The other thing that we could show is that assembly at the tip required an active transport, so an energy-requiring transport step called intraflagellar transport, which is required to take the protein which is made in the cell body out to the tip where it can be assembled. And I'm going to just show you a little bit about this intraflagellar transport process because it ends up being critical to our model.
It seems like a waste of energy to assemble, disassemble, assemble, disassemble. I mean, you would think that the cell would optimize somehow
yes.
energy.
It's a great point. And actually some cells don't do this. So in mammalian sperm, they start off doing this constant turnover, and then once the sperm have reached their final length, they turn off all their turnover and they lock the length down. And this is also true in, if you know about ciliates that live in the pond, like Paramecium, Tetrahymena. When their cilia first grow, they're dynamic like this, and then once they've reached final length, at some point they just lock down all the dynamics and they stop doing the turnover. So why does Carbonimonas keep doing it? I believe it's because in order to do a proper breaststroke motion, you want both of your flagella to be equal length. And I'm going to show you the model for what we think controls length. And when I get to that model, so I'm going to show you a graph with two curves on it. When I get to that point, I want to revisit your question. And if I forget to, please stop me, because I think we have an answer to that actually. And it may actually be for a smart reason. So this transport process, what you have is you have a motor protein kinesin. So this is a protein that burns up energy. It hydrolyzes ATP, which is the cellular energy source, and uses that energy to move along these protein tubes, the microtubules. So the kinesin molecule moves along the microtubules from the cell body all the way out to the tip. It attaches itself to a spherical ball of protein called an IFT particle. It's got about 20 different proteins in it. This IFT particle, these proteins are not part of the flagellar structure. They're more like the bed of a flatbed truck. That is, they're something that you can put cargo on and then have it be carried out. So for example, the protein molecules that make up these microtubules will actually bind to these IFT particles and then be carried out to their site of assembly. And just a question of terminology, because it ends up being important, these IFT particles form linear arrays, which we call IFT trains. So it's like the cars of a train. And this whole train will move from the cell body all the way out to the tip, drop off its cargo, and then turn around and come all the way back. And there's another motor called cytoplasmic dynein that brings the train back. So there's constant motion going on back and forth, presumably for the main purpose of supporting this ongoing turnover, which is already discussed, seems wasteful anyway. Okay. So now we have this picture where we have, if we want to see, what controls the length of these protein tubes, microtubules, and we know there's constant assembly and disassembly, and the assembly requires transport. So if you're going to take the system and then build a control system onto it, whatever kind of system you build, its goal has to be to make sure that these two rates equal each other, if and only if you're at the correct length. If you're too short, you want assembly to predominate so you can get out to the right length. And if for some reason you got too long, you would like disassembly to predominate so you would shorten back to the correct length. So this is the goal. So the question is, what do we have to add onto the system to achieve this condition? A couple of facts that we found along the way is that this rate of disassembly is apparently independent of the length. So if you have a long flagellum or a short flagellum, it's pulling off protein at the same rate. So therefore, in order to achieve this condition, you'd have to be controlling assembly as a function of length. And we know that assembly is limited by transport. So if we use mutations that increase or decrease the rate of transport, we see a concomitant change in the rate of assembly. So that raises the question of maybe this transport, interflagellar transport, or IFT, could that itself be length regulated? And could that be a way that you're actually achieving this condition? So the question becomes, in what way is IFT, this transport process, regulated?
Just a
Question, yeah.
dumb question.
No, that's
do we know why the growth doesn't take place at the root instead of at the tip?
Yeah, so I'm simplifying this by just showing a line, but actually what you have here is this nine-fold symmetric array of these tubes. And the reason why you have this beautiful symmetric nine-fold pattern is because there's a structure inside the cell, which is the centriole, which was able to self-assemble in nine-fold symmetry. And then the tubes inside the centriole nucleate these tubes in the flagellum. And so it grows off of this structure that's already there. So there is not really anywhere that you could add any new subunits in. Now, I assumed at first that actually the turnover was going on at the base. And I had a big fight with my advisor, and we bet a case of wine. He ended up being right, and I never paid up because we couldn't agree on which kind of wine I was going to. So, okay, so right. So the question. So how could this transport be regulated? Now, one thing we know is that once these particles get in, their fate is determined. So they're going to move all the way to the end. They never stop. They never fall off, and they move at pretty much a constant speed. So if you want to control this transport, you have to control the transport at the stage in which the particles are brought into the flagellum from the cell body. So the question becomes, what controls how much of this IFT protein gets in? And we know that it's not just that you use them all up, there's always a pool of these proteins in the cell body that are not being used at any point in time. So what determines how many of them get in? How does the cell know how much to put in? Now, I always believe that maybe the cell doesn't control it. Maybe when you first build your flagellum, you load it up with a certain number of these particles, then they just circulate back and forth. And we can talk about it later, but if this is true, you can make a really beautiful, simple model for length control that's very satisfying and nice. But it turns out it's not true. I mean, I always believe this, and so all my students set their aim at trying to prove me wrong, and they succeeded. Okay, so this doesn't work. And one of several ways we've shown it doesn't work is by using this method called fluorescence recovery after photobleaching. So what you do is we put a fluorescent molecule onto these IFT particles, and then send them in. So when we look in living cells, so we see these little fluorescent particles moving back and forth in the flagellum. And then we photobleach, so we shine a very bright laser which bleaches all these molecules, right? So if they never go in and out, once you've bleached all the fluorophores, all the fluorescent molecules in the flagellum, it would just stay dark. Whereas if they can exchange for new ones, now they'll exchange with new ones from the cell body that are still fluorescent, and you'll see recovery of fluorescence. And I have some movies that I could show you. I wasn't able to figure out how to insert the movies into PowerPoint, sorry. But so what we can do, this was done by my postdoc Hiro Ishikawa with a team of students at the Marine Biology Lab at Woods Hole. What we could show is that you do in fact get recovery of fluorescence on the time scale of tens of seconds, and actually the recovery that you get happens at exactly the rate that you'd predict if every single particle that goes in, goes to the tip, goes all the way back, and then comes out again. And we've now verified this in an opposite way. So Hiro has made a different kind of fluorescent tag where you can turn on the fluorescence once it's in the flagellum. And so you make these things become fluorescent red, and you can see them coming out and moving into the other flagellum. So we know for sure that there's not a barrier keeping everything in there. So now we do have this question, though, of what is controlling how many of these proteins get in. So we wanted to get a better look at this insertion of protein into the flagellum. And so my student Will Luddington wrote some very nice software to, I won't really go through just in interest of time, but ways where you image at high resolution these particles moving from the cell body into the flagellum. And then he had a way to convert the images that we get into a time series trace where you have a series of peaks where the area under a peak is telling you how much protein is going in per injection event. So how many of these particles, how big is the train that's going in at each point in time. We learned a lot of cool things from this. So it turns out that these time series traces have a lot of the same features as studies of earthquakes. So you have this correlation between how long it is before an event and how big the event is, and also between how big the event is and how long it is till the next event. And we also see bursting and quasi-periodicity. So we think it's some kind of an avalanching system, although we don't know exactly what it is that's avalanching. So that's just a sort of an empirical observation question.
The growth curve that you showed us, that was a single flagellum.
Yes.
Was the other one cut or growing, or was it, or were there any other attached to that cell?
It's a great question. So in those experiments, typically you chop off both flagella. You can, and I have slides to show you this, you can chop off just one of them, and you can do that. The previous way was you would push on the cover slip with your thumb until you hit it just the right amount of force, and one of them would pop off. But now we do it with lasers, where you just ablate one of them. And the great thing is, if you pop both of them off, they grow back like I showed you. If you pop one of them off, what happens is actually, I think I do have a slide on this at the end. Is that okay if I just,
Sure.
sorry, I'm giving away, I may not even get this other story. I know.
Oh, now I see.
Oh, I don't. Okay, okay. Now I'm really. Anyway, so let me go back to where I was while I answer your question. So if you puff up one of them, this is the result first found by Joel Rosenbaum. Again, as the one that you popped off grows back, the other one starts to shorten as though it knows it's too long, and that will keep happening until the two flagella reach the same length, and then they grow back out together. And I think, and this gets. And so I think that's, so no matter what you do, the flagella want to be at equal lengths. And I think the only way you can do that is to have this constant turnover. So I'll show you them in a second, I'll show you, I think, why that happens. But I think teleologically, the reason why the cell wants to do that is to ensure that you're always able to do a breaststroke. Because if you have unequal lengths, you'll spin in a circle. And we have movies showing that happens, actually. Okay, so, all right, so we can measure these traces. And one of the things we can measure is just, what is the rate at which IFT proteins are entering the flagella per unit time? So how many of these particles are going in at any given point in time? And when we plot that injection rate versus length, it turns out to scale as roughly 1 over the length. Okay? So that means that somehow length is controlling the injection. Now, going back to this picture, we say that disassembly is happening at some constant rate, assembly is happening at a rate that's determined by transport, and transport is scaling as 1 over the length. So now we can make a really simple model, which is shown here. So now I'm plotting rates versus length. The rate of disassembly is length independent, so it's a flat line. The rate of assembly is going to be going as 1 over the length, so you have a hyperbola. And there's only one length where those two curves will intersect, and that's your steady state solution. And you can see that it's stable. If you get too long, disassembly is higher than assembly, so you'll shorten. And if you're too short, assembly will predominate, and you'll grow back to that length. So this can explain why you're able to achieve a particular fixed length. It also explains exactly the kinetics of flagellar regeneration, which, as I said at the beginning, the rate of flagellar growth is going as 1 over the length, and I think this explains why. It can also now explain this issue of why do the two flagella equalize their length. Because imagine both flagella are sort of sitting at this operating point. Now you make one of them much shorter by chopping it off and letting it grow back. Now it's going to be an operating point over here, where assembly is much, much faster than disassembly. So when they're at this operating point, they're growing and disassembling at the exact same rate, so they're sort of protein neutral. They're turning back whatever it is they used up. Whereas when they're very short, now they're net consumers of protein, and so they're taking more than their fair share, and therefore they're going to starve the other one for protein. And so because it was balanced right at this point, when its supply of protein goes down, it's going to have to shorten. And so I think this is the way you achieve equalization of lengths, and we can build models based on measured parameters, and we can exactly match the kinetics that we see in the actual cell.
Maybe one reason my earlier question about why this inefficiency of assembly disassembly is that it's constantly trying to adjust to get in the same length.
Exactly, because if you didn't, right, so, I mean, this whole model requires that disassembly has to constantly be going on. If you didn't have this, then it would just be whoever started growing first would win, and you wouldn't be able to equalize your lengths. So, and also from all of our imaging of cells moving, if you make both flagella longer or both flagella shorter, if you do it at a very extreme case, you get these Reynolds number effects where the cell can't swim, but small effects don't make much difference. But if you make them even a little bit unequal in length, then they can't go in a straight line and they can't find the light source. So we think that these cells, that their main fitness criterion is making equal length flagella, and to do that, it's exactly what you say, you have to have this constant turnover, and then that allows this kind of mechanism to equalize their length. So I always talk about this as a length control, as though the goal is to achieve a certain length. I now believe the goal is to achieve equal lengths of the two flagella. That's the primary goal. Okay, so this model I'm pretty happy with. I feel that it can explain all the known phenomenology. For example, we can predict very nicely the dependence of the length of the flagella on the number. So if you add more and more of them, which we can do using mutants, they get shorter and shorter, and they get shorter and shorter in a way that's exactly matched by the predictions of this model. So it's great because it can explain everything. Unfortunately, it also doesn't actually explain anything, because all we've done is change the problem. Because now the question is, how does the cell regulate injection of IFT? So ultimately, it's the 1 over length dependence of IFT injection that's achieving all of what we're seeing. Where does that come from? So we have a new problem now, which is how does the cell, A, measure how long its flagellum is, and how does it use that information to control the entry of these particles into the flagellum? And at this point, we really don't know how this works at all. So now we're at a stage of trying to come up with possible models and find ways to test them. And this is, I think, a fun thought experiment. Think of, if all you can play with is molecules that can move and so on, can you build a system that can measure length on the size scale of tens of microns, which is very, very large for a single molecule? And it reminds me a lot of the famous experiment, the famous question, which is how many ways can you measure the height of the Empire State Building using a barometer, right? You guys have probably done this. One is you go to the top and you measure the barometric pressure difference. But the other way is you drop it off the top and time the fall of the barometer. And you can give the barometer to the building manager and say, tell me how tall the building is if I give you this barometer. You know? So we're trying to think, what are all the possible ways you can imagine that you could use molecules, proteins, to measure the size of something which is 10 microns long?
Is the mobility rate constant?
The mobility rate of the motors, it's slight.
Of the molecule that moves.
Yeah, I'll show you a picture in a second. It is slightly dependent on length, but it's very, very slightly. So we think it's more or less constant, yeah. Which leads to actually one of our models, actually. This maybe is what you're already thinking. So one way you can measure a distance, if you already have a train, this is sort of an Einstein thought experiment. If you put a clock on the train, let the train go out and come back, and you know how fast the train was going, the time elapsed on the clock tells you how far the train traveled, right? So one model is we call the time of flight model, which my colleague Bill Snell at Southwestern first proposed, to my knowledge, first proposed it. Is you put a molecular timer onto one of these IFT particles, send it out at a speed that's more or less length independent, let it go out, come back, and read off how much time has elapsed. And that will be a proxy for the distance that it's traveled. And the nice thing is that we know a lot about how molecules can act as clocks. So there's the whole class of molecules called GTPases, which take a small molecule called GTP, guanosine triphosphate, and they convert it to another molecule, guanosine diphosphate. And this happens with a very definable time scale. And so what you can do is, if you load up a whole bunch of these things with GTP, send them all out, and then ask how many of them still have GTP or how many of them have turned it over to GDP, that ratio tells you something about how long they were in the system. And it turns out 2 of the molecules, I remember mentioning that there are about 20 or so proteins in these IFT particles. 2 of them actually are these GTPases that are known to act as timers in other contexts. So there's at least 2 candidate timers already in the system. But this is only one possible way to make a timer molecularly. Other ways are you can have any kind of modification of these proteins. You could have proteins that get phosphates put on them, and you have maybe some enzymes sitting here that are putting those phosphates on at a certain rate. So the longer they're inside the flagellum, the more phosphates they would get, and you could use that as a readout. So the question is there a way to test this kind of model without having to guess the identity of the timer? So can we either rule in or out this entire class of molecules? Okay. All right, great. So one way we've been doing this is we actually did an entire genetic screen just to get a reagent to test this model. So what we screened for was proteins with mutations that would slow down the rate by which these particles come back to the cell body. Right? So they go out just fine, but now they're slower returning. So it's a mutation in the motor that brings the trains back. As a result, they're traveling more slowly on the return trip. Okay, so the idea is that if you can slow down these particles as they come back, that means that they're spending more time inside the flagellum. So for any given actual length, the cell, if the cell is using this kind of timer mechanism, it should think that the flagellum is longer than it actually is. So the test is, look in this mutant and ask, does the cell think the flagellum is longer than it is? Now the question is, how do you know what the cell is thinking?
Disassembles.
Well, kind of, but that happens, yes, so you could try to ask that and ask, can you make would the flagellum get shorter than it's supposed to be? And let me return to that point in a second. So that's one way that we thought of. The other way, though, that we tried was to look at the IFT injection process itself, because what we knew was that as the flagellum gets longer, it's not just that so several things happen. The reason why, remember I said how the size of the injections, the rate of injection goes 1 over the length. But actually 2 things are happening that feed into that. One is that the trains get smaller and smaller. So there's these linear arrays of IFT particles, and as the flagellum gets longer, there are fewer and fewer train cars per train. So that's illustrated here. So the trains get smaller and smaller as the length increases. But the frequency with which trains go in goes up a little bit. So the difference between a long and short flagellum, if you just look at IFT particles going in, is that a long flagellum will have a high frequency of small trains, and a short flagellum will have a low frequency of large trains. So the question is, so the idea is, the prediction of time of flight model is if we slow down return using our mutant, what we should see is a high frequency of small trains. And in fact, what we see, and this is just images where we've taken line scans through the flagella and stacked them up, so you see these diagonal traces are individual particles moving from the base out to the tip. What we see is that compared to wild-type cells shown here, the mutants have bigger trains than they have less of them, which is exactly what you see in a short flagellum, not a long flagellum. And they also start to get short, as you suggested. So in every respect, it looks as though the flagellum is behaving like it's shorter than it wants to be, not longer. So it's still responding to the change, so maybe it's trying to compensate for a defect in the transport, but it definitely does not fit the time of flight model. So we believe that we can rule out that entire class of models without having to go and test all the different possible molecular timers that might exist. That's nice because we've now sort of cut off one whole branch of the tree of possibilities. But now we're left with, well, what is it? And one model that I really like is the idea that maybe something, let's see if I don't have a picture of this, but one model would be maybe out at the tip, you have a source of some diffusible molecule that then has to get back to the base to send some kind of a signal. And the idea would be that as you got farther and farther away, if the molecule was being generated in pulses, it would maybe the time required for the pulse or the size of the pulse would go down. Or if it's constantly being made, you'd reach some kind of quasi-steady state and have a linear gradient of concentration, which you can then read off somehow as a proxy for length. And we actually have a candidate now. So I mentioned that there's this particle of proteins, the FT particle, that's the bed of the truck. But then there's the motor, the kinesin motor, that moves out and brings them out. And it turns out that motor, so the actual particle itself moves processively out, so it moves without stopping in a very linear way, and it moves back with another motor. Whereas the kinesin is not brought back by that motor, it appears to diffuse back. So I think there's maybe a very elegant solution here where the kinesin motor itself that's driving the entry of these trains has to diffuse back in order to pick up another load of trains. And that could actually set a length scale. And so now in order to test that though, we need ways to modulate the diffusion of something. And we have lots of tools in molecular biology to change the function of enzymes. We have very few tools to change the diffusion constant of enzymes, other than just sticking on great big chunks of garbage, which can have other unintended effects. So now we're sort of at that stage with that problem. Yeah.
Well, we're still there. How much do we know about the interaction between the moving entity and the surface of the core?
And that's a great.
Either its topology or its chemistry.
So the surface of so the outside of the flagellum or the.
Yeah.
So
both of these passages mean that stuff is passing by the core, and if the core is poorly done, or if it starts to develop defects or something, change the rate of movement.
Yeah. So right. So there's a couple of things actually. Let's go back to the IMT cartoon. So there's a couple of issues actually. So these particles, they're linked to the protein core through the motor, but they're also directly attached to the membrane via chemical linkages. And so if you do things that affect, for example, the ability of proteins to move on the surface, you can actually jam up these trains.
Yes.
And so one thing that happens is if you put these cells down on a glass slide, and actually these cells, I should say, we talk about them swimming in a pond, mostly though they live in the dirt. And so their typical environment is actually the surface of a dirt particle, which at this size scale is basically a flat surface. So they'll put their two flagella down in opposite directions, and then as these trains are moving, sometimes they'll get stuck on something on the surface, and then they'll pile up there until they generate enough force, and then that entire thing will start to move, and it will drag the entire cell body. And so cells can actually glide over a surface using this motor. And so, so there's an interaction, but then eventually this becomes so powerful that the entire cell just lets go and moves. And the interesting thing is the two flagella are trying to move in opposite directions because their motors are running in opposite ways, right? And so there's a tug of war between them. And we've imaged this a lot, and so it turns out, I said how when cells are swimming, they want their flagella to be equal in length. When they're gliding though, if they're equal in length, they tend to just jitter back and forth, they never get anywhere. Only if they're unequal in length can they now move in one direction. And so I kind of think that maybe there's a trade-off between what the cell wants to do. Does it want to, if it makes its flagella too equal in length, then it's great for swimming, but it can't do gliding. If they're too unequal in length, it can glide, but it can't swim. So maybe there's some trade-off in this. And cells grown under different conditions actually have different levels of equalization of their lengths. And the cells that are most equal in length are cells that are supposed to be swimming and finding each other and mating. So they're basically just for swimming, and they have incredibly highly equal length flagella. Regular cells that are living out in the dirt have somewhat unequal lengths, and I'm thinking maybe that's for an actual reason. Maybe there's the cell is deliberately breaking what could be a perfect control system and making it imperfect for actual biological reasons.
Is that movement on the dirt a feature rather than a bug?
It's a good. I'm going to say it's probably a feature, and in fact, it might. So at the very least, it will allow these cells to disperse. So these cells are very good at dividing if they have enough nutrients, but eventually they don't want to just pile up in a little ball. They want to spread out. And so it absolutely lets them spread. So you can. So Keith Kosminski, who used to be in the Rosenbaum lab, did this experiment where if you put these cells on the surface of a petri dish, they will eventually spread out over the surface, but only if they can do the gliding motion. So it's at least a dispersal mechanism, which tends to be important in colonizing the environment. Also, I'm not so sure whether it's true in these cells, but other cell types use this gliding motion to bring food to the cell body, and this is thought to be why these structures first evolved. So the intelligent design people always tell you, well, in order to make this thing that can actually bend and swim, there's so many things that have to evolve at the exact same time, it's impossible. Whereas if maybe what they first did was they were just trying to make some long thing that could do the surface motility, and for that you don't need all this fancy coordination of all these bending motors. So the current thinking is that might be the original evolutionary reason why the structure was formed. So I'm assuming it's a feature, not a bug, but yeah, who knows. Question in back.
So the growth rate is proportional to the arrival rate of the IFT particles. So the number of IFT particles for any length flagellum is about the same. So is this just restating the problem? So that it sort of says that the flagellum is allocated some fixed number of IFT particles. And so there could be some mechanism back at the root that's saying, well, you have enough IFT particles right now. You don't get any more yet.
Yes. So, and in fact, so what you said, and you are not by any means restating it, but it's what I always see. The way we first found this was by finding what you said first. But the first thing we saw was that the number of these particles was fixed, independent of length. And then the question was, well, how do you explain that? And the original model I had was maybe, as you say, you're initially allocated a fixed number of them, and then they're locked in there. Now we know they aren't locked in there. Nevertheless, it's still possible that you could allocate an initial number and then have some kind of a gate here that lets a new one in every time an old one exits, right? And so I think that would absolutely work. And so one thing that we're trying to do now is look very carefully at the relation between the return of a particle and the entry of another particle. Is there any coordination between those? And we didn't have good tools for doing that, but now we do. So I think that sort of ping pong mechanism could absolutely still work. Yep, I like that idea a lot, actually. I still want to believe there's some kind of pre-allocation because it would solve so many problems. You wouldn't have to measure length at all then, right? And the beauty is, so just going back to this model, suppose that either there's a fixed number that is locked in there, or there's an initial fixed number, then there's some kind of ping pong, exit is coupled to entry. Then it will naturally be the case that the rate at which they leave will scale as 1 over the length, because when you're twice as far, the round trip transit time doubles, which is what you're getting at. Yeah, very perceptive. Question?
So you've told us that these cells are sensitive to fluid flow, and so they must have some sort of mechanical response. There would also be a change in the mechanics associated with the viscosity and the length of the flagellum. So is that a possible feedback mechanism?
I think so. Yeah. So the question is whether it's sensing things like viscosity ultimately by sensing fluid flow, or it could just be sensing its inability to bend as a function. It could be sensing resistance to the bend. We don't really. What I would love to have is a nice way to tune the viscosity of the media. And the frustrating thing is that most of them, we need to do this better. The best people typically try like adding things like sucrose or something, change viscosity, but they also change the osmotic conditions. And the problem is that if you add a lot of solutes to the external environment, you end up sucking water out of the flagellum, and you end up making the interior more crowded. And you can actually see these trains grind to a halt when you do that. So I think we need to find a way to put some kind of filaments or something out here that impede the motion without having an osmotic effect, and I think that would be very interesting. Or the best thing would be if we could actually put magnetic beads on this thing and actually force it to bend in a way of our choosing. Does that mean 5 minutes over?
5 minutes left.
Okay.
Well, you can go over it.
Well,
I don't mind, but just to give you an idea.
I mean, I'm basically done with this story. I mean,
You can show us the movies.
yeah, we can see look at the movies on my computer. Should we just do that?
Show us the movies.
Well, any more questions before we do that?
So just the relationship between shape on this level and shape on the kind of the next level. Like, is there any kind of are there any analogous processes on in the two cases?
Yeah. So for example, at the level of the entire cell.
Well, I mean, versus the liver has some shape.
Oh yeah, right. So I would like to believe that would be the case. And actually, the other story that I had, which I'm not going to actually tell you, but I'll just show you, is we're looking at this cell that's 1 millimeter long. So you're having a cell that's now at the same size scale as an embryo, and I have a whole argument for why I think this is doing all the same processes that an embryo does when it develops. So my personal sort of, I guess, religious type belief is that multicellular things like organs or animals are essentially doing the exact same biology that individual cells do. So I would like to believe that the lessons we learned from an organelle might apply to things like the liver. People tend to think about organs as having a lot more sort of supervisory monitoring by the rest of your body. So there could be things within some gland that are sensing how much your liver is giving output. But people don't generally know where the shape comes from. The clearest example where the shape of an organ has been carefully, really, I think, explained is Mahadevan at Harvard has been doing the study of gut looping. So the gut, it's not just a straight line, it loops in a certain way. And it has a characteristic wavelength that different species will have a certain wavelength of looping in their gut. And his model, which they've now tested extensively, is is it okay? So the gut is a tube of cells, right? So there's a tube of cells that grows, and the whole thing is growing. But the gut is not just hanging out free, it's attached to the body wall by a sheet of cells called the mesentery. So you have this tube growing and a sheet that's holding it onto the body wall. And the rate at which the tube grows is different than the rate at which the sheet grows. And the tube grows faster than the sheet, so the only way that can happen, but the sheet is rigidly attached to the tube, so the only way for that to work is the tube has to bend so as to accommodate a given length of the sheet. And they built models out of rubber that replicate that, and you can go into, for example, into an animal and cut the mesentery, and then you can lose the looping, and you can predict the wavelength of the tube exactly from the elastic properties of the mesentery. So that, I think, is probably an open and shut case. So that's quite different than this. That's much more mechanical, right? And so, and I think that probably the shapes, I'm focusing on the size of this one organelle. And as mentioned earlier, this is a case where the cross-section is invariant. So the shape is not really allowed to change other than the length. This may be a very specialized case. Most organelles are a lot more flexible, and they can, if you put more membrane on, they can change shape in various ways. And there, the shape does tend to, I think the current, wherever it's been studied, it appears to be largely mechanical. So you're trying to minimize the total energy from all the bending energies and so on. So I think there will be probably good analogies between overall organ shape and organelle shape. For those, I think we've picked the worst one for doing what you want to do. Question?
Again, a very simple question. We haven't talked much about what happens at the interface when these cells are resting on another surface, a different surface. And there's chemistry that changes the internal organization of that cell, so as to change the pipeline that goes into the organelle to these.
Absolutely. Yes.
Yes.
Yeah. So we've been looking for example. So Cleminomonas, when you put them on a surface, they'll glide for a while. But if you leave them on a surface, they will actually pull their flagella in.
Yes.
And then grow without them. And then if you wash them off into water, they'll grow their flagella back in about 10 minutes.
Yeah.
So they somehow know whether they are or are not on a surface. And what I would like to know is how, if you pulled your flagella in and they're surrounded by a apparently rigid cell wall, how do they know whether they are or are not in contact with a surface at that point? And this has been studied now for bacteria. There's some very nice studies of how bacteria can sense their surface. And so I think we don't know for our cells, but they somehow know. And we've also looked at mammalian cells, and you can grow them, normally you grow them on a glass dish, but you can coat that glass dish with polymers of various stiffnesses. And then if you look at things like the mitochondria or the nucleus, the size and shape of all the organelles actually changes as a function of the stiffness of the surface.
Yes.
So it's exactly what you say. There's I think it's both chemistry and mechanics
and
are feeding in.
Hydrophobicity
and all these things.
Of the surface.
Exactly. All these things I think are feeding into that.
Yeah.
And so I think it means you need to think a lot about what kind of surface chemistry and properties you want if you're going to grow cells on something.
Yeah.
But it also means the flip side is that could be a way to provide input into the cell, right? So you can put a cell on something where you could dynamically change the stiffness or stretch it. That could be a new way to tell the cell what to do.
Probably also makes experiments somewhat more difficult because you can't just drop it on there and assume that
you've got to be very careful.
The support didn't change.
It does, and it creates a lot of fights actually. And there's so one fight is, well, what is a real surface? Is it just glass? Obviously, there's no glass in your body. So then, well, you coat the glass with some protein like fibronectin that's found in your body. But in your body, it's not just there as an amorphous sheet. It's typically woven into filaments. And so I think people are trying to make more and more lifelike systems. Then the question is, well, is a flat surface even the appropriate context anyway, right? Some cells do, in fact, live on a flat surface, but others live in a three-dimensional gel. And then you want to put your cells into that. And then the problem is they typically, instead of spreading out nice and flat, they form these balls that you can't see anything anymore. So it's a huge issue right now. And almost whatever anyone does, someone else says, no, that's not the right thing. So then maybe the best way to do it is to actually go inside a living organism and study the cells there. And so there, the technology's gotten amazing. For example, in living mice, you can go in and watch every cell dividing, as the lung develops, for example. A colleague of mine does this, and it's really beautiful what you can do. So that's part of why I love these free-living cells from the dirt of the pond, where there's no issue about their context. We know they're living on hard glass-like surfaces and in liquid like you find in a flask. So I feel a lot better about free-living things. And Also, a cell in your body is always talking to other cells. And so even if you give it the correct physical input, it may not be getting the appropriate signals from its neighbors that it would normally be seeing. And so my personal preference is, I think that's a really, that's both a bug and a feature, right? I mean, that's very interesting to study, but we try to avoid that by always going with these free living things whenever possible.
Yes.
Movies.
So let me make a proposition.
All right.
How about we go to the conclusions so that we can close the web meeting, and for whoever is interested, we can show the movie.
Okay, sounds good. And I have a whole other talk about this giant cell that develops like an embryo, but that's... Believe me, I would much rather have great discussion about one story than just have no one say anything and go through two stories. So I just want to close by acknowledging our very generous funding from NIH especially, and also now from NSF, and a huge list of collaborators, only a small fraction of whom are shown here. And then current collaborators, including Simone, and then amazing students and postdocs from my lab who've actually done everything that I'm showing you. So if we have any more questions, we can talk about that. Otherwise, then we can go look at some movies.